audio recording of lecture 4, virus replication cycle. Slide number 2. In cell culture, the viral replication, meaning that making more of itself, is a one-step process, and we have high level of virus infection. 1 to 10 virus can infect a cell and they undergo replication themselves. The replication that these viruses undergo are synchronous replication, and all of these events that we see in cell culture are required for in vivo infection of the host cell by viruses. Virus replication cycle has several different steps. The very first step is the attachment or adsorption. Virus attaches through a cell surface specific receptor on the host cell, and through this adsorption or attachment, virus can gain entry into the cell, or so-called penetration, and after that there would be dissociation of viral capsid from its genome. The second step during viral replication cycle is early gene expression. We have discussed this before. Through the production of early messenger RNA and translation of these messenger RNAs, viral proteins will be made. These are usually non-structural proteins required for the next step or viral replication. These can be enzymes, polymerases, etc. Genome replication is when the genome of the virus makes more of itself, and this step is followed by late gene expression due to the production of late messenger RNA and translation of these late messenger RNAs, and usually they make structural proteins that are involved in the formation of the protein shell or capsid of the virus. At this stage, we have structural and non-structural proteins being made. Genome is being replicated. So the last stage is assembly or maturation of the virus. All of these components that are being formed will assemble together. The virus is mature. It's called a virion. And at this stage, virus will be released from the infected cell, as shown at the bottom of the slide here. This release of the virus from the initial virally infected cell can be accomplished through two mechanisms. It's either lysis, where the cell will burst, or through budding. The virus chooses a specific region of the host cell plasma membrane and exits through the host cell plasma membrane. So let's talk a little bit about virus attachment and adsorption, slide number 4. Initially, there is a contact and interaction between the virus and host cell. This interaction is very unique and very specific. So virus needs to need to be recognized by the host cell, and this is done through the interaction of virus with specific surface structures called receptors. Those molecules of the virus that bind to host cell are called ligands. So ligands will interact with receptors. And ligands are usually made up of viral proteins or viral glycoproteins, those proteins that have undergone glycosylation and a sugar moiety is being attached to them. Slide number 5, virus binds to host cell molecule called receptor. And a receptor is a host cell protein, or it can be a glycoprotein or a lipid. Receptors usually uh, have a role in normal functioning of the host cell. We'll talk about some examples. You will see that, for example, a receptor called ICAM1 is actually an adhesion molecule. So they have a role in normal cellular functioning, but viruses can also recognize and bind to them. So, slide number 6, a few words about cell receptor. Viruses can bind up to 3 different types of receptors in succession. Initially, they bind to low affinity receptors that are in high abundance. There are lots of them. And virus contacts the cell surface through these low affinity receptors. The second type of virus is primary receptor, which in contrast to low affinity receptor has lower concentration on host cell surface. And lastly, there is co-receptor, that binding of the viral ligands to co-receptor follows binding of the primary receptor. So these 3 types of primary receptors are low affinity receptor, primary receptor, and co-receptor. Attachment of the virus is very specific. The specificity of the attachment is discussed in slide number 7. In this respect, we talk about 2 concepts. One is host range, and the other one is tissue tropism. Host range is basically the organism or organisms that the virus is able to infect. It can be a plant, it can be animal, or it can be a human. And viruses have either narrow host range or wide host range, meaning that they can infect a plethora of hosts. The other concept regarding specificity comes from tissue tropism. Tissue tropism is basically the cell type or cell types that a virus is capable of infecting. These can be either skin, oral mucosal cells, gastrointestinal tract cells, or central nervous system cells. So these 2 concepts, host range and tissue tropism, contribute to the specificity of the attachment of virus to the host cell. Slide number 8 talks about another aspect of attachment, which is the binding itself. So the interaction between viral ligand and host cell receptor follows basically a 3-dimensional feat, and it's usually uh, accomplished by weak electrostatic charges. And the evidence for these electrostatic charges uh, uh, come from the fact that this interaction requires a very specific receptor, if we specific pH, sorry. If we modify the pH, the binding may not occur. Specific ionic strength, as well as the presence of specific ions such as calcium and magnesium that facilitate the viral ligand and host cell receptor interactions. Slide number 9 is a specific example of the attachment of a non-enveloped virus called picornavirus. Picornavirus has a ligand, and this ligand is basically embedded within a deep cleft or canyon in the triangular face of capsid. Three capsid proteins, VP1, VP2, VP3, come together, they form a cleft, and this is basically the ligand. And this ligand interacts with a host cell receptor called ICAM1. And ICAM1, as I mentioned previously, stands for intracellular adhesion molecule, and ICAM1 participates in the binding of it's expressed by white blood cells, so its normal function is to allow the white blood cells either to bind together or bind to the endothelial layer of the host cell. Slide number 11. Let's talk about attachment of an enveloped virus such as HIV, human immunodeficiency virus. There is a host cell protein that is within the virus envelope called cyclophilin A. It initially binds to HIV, to low affinity receptor heparin sulfate of the cell. So the very initial step is the binding of cyclophilin A to heparin sulfate, as shown at the bottom of the slide. Following this interaction, remember this is a low affinity receptor, heparin sulfate, so it's going to be in abundance. Following this binding, a viral ligand called GP120 binds to host cell receptor called CD4 on the surface of helper T cells, macrophages, and glial cells. Remember, heparin sulfate is a low affinity receptor, CD4 is a primary receptor. Upon binding of GP120 of virus to CD4 on the surface of host cell, there would be a conformational change in the GP120. After this conformational change, GP120 binds to two other receptors on two different cell types. It can either bind to chemokine core receptor called CXCR4 expressed on T lymphocytes, or it can bind to another co-receptor called CCR5 that is expressed on macrophages. So the steps are cyclophilin A binds to heparin sulfate, low affinity receptor. Heparin sulfate is a low affinity receptor expressed on the surface of host cells. Upon this binding, there would be the binding of viral ligand GP120 to a primary receptor called CD4 that is expressed on T helper cells, macrophages, and glial cells. This binding results in a conformational change of GP120, which allows GP120 to bind to two different types of chemokine co-receptors on the surface of T lymphocytes called CXCR5 or on the surface of macrophages called CCR5. Slide number 13, entry and uncoding. Entry is the mechanism that is being used by the virus in order to penetrate into the host cell. That's called penetration. Mechanism used by the virus to gain entry into the host cell. Another process is called uncoding. Uncoding is basically the separation of viral nucleic acid from its capsid. Uncoding refers to those changes that happen and make the viral nucleic acid for expression. As a matter of fact, this is a collective term. It makes the viral nucleic acid for gene expression as well as its replication. We'll discuss these in future lectures. So let's talk about a couple different scenarios of entry and uncoding of a non-enveloped virus, a virus that is so-called naked. It happens through receptor-mediated endocytosis using clathrin-coated pits. Clathrins can be seen by electron microscopy. These are small proteins. So basically, virion or viral particle recognizes specific receptors on host cell plasma membrane and a region of the host cell plasma membrane that in the inner leaflet, there are these proteins called clatrins are sitting, virus binds there, and then the next step would be invagination. This clatrin-coated pits that encompasses virus that has been interacted through its ligand with host cell receptor, in this case ICAM receptor, will be invaginated and will get into this host cell cytoplasm. Once this clathrin-coated pit gets into the cytoplasm, inside of the cell forms an intracellular endosome. That intracellular endosome contains the virion or viral particle. And in an ATP dependent manner, hydrogen ions will be pumped into this endosome and endosome becomes acidified. So let's talk about uncoding now. Virus is in endosome. Endosome has an acidic pH. This acidic pH will cause a conformational change in the viral capsid proteins in a way that the hydrophobic regions of these viral capsid proteins will interact with the membrane of the endosome, create a pore. Through this pore, the viral nucleic acid will be released into the host cell cytoplasm. Slide number 18. So far, we discussed the entry and uncoding of a non-enveloped virus such as poliovirus. Now let's talk about the entry and uncoding of an enveloped virus. Enveloped viruses are those viruses that when they are budding or coming out of the host cell, they pick up a piece of the host cell plasma membrane. So entry is through receptor-mediated fusion of virus envelope with host cell plasma membrane. As you know, both are the same composition, viral envelope and host cell plasma membrane. And then there are two ways after this initial fusion, there are two ways that virus can gain entry or penetrate. One is direct, and this direct is basically pH independent. It does not require a change in the pH, no endosomes, and as shown in the left hand side of the figure, where the capsid of the virus will be released into the host cell. And at this time, the genome of the viral host cell will be released inside of the cell. You will see a term called nucleocapsid. This is the combination of the nucleic acid of the virus and capsid. Another pathway that enveloped viruses can use is called receptor mediated endocytosis, and binding to a specific receptor again, and the formation of endocytic vesicles pinches off or invaginates. It will be the endosome will get into the host cell cytoplasm, acidification of the endosome, conformational change in the capsid protein, creation of a pore in the endosomal membrane through which viral nucleic acid will be released inside of the host cell. Slide number 19. The direct entry and uncoding of an enveloped virus such as Sendai virus. So the virus will bind to a very specific receptor at the cell surface, and this will result in the fusion of viral envelope and host cell plasma membrane through which the viral genome will be released into the host cell cytoplasm. There would, upon this fusion, there would be a conformational change in a viral fusion protein, that this viral fusion protein becomes active upon cleavage. This is the fusion peptide, as you can see. It's a change in its conformation, and then after that, virus gets into the host cell cytoplasm. Slide number 21. Uh, Another example of entry through receptor mediated endocytosis of an enveloped virus such as influenza virus. As we talked, the pH in the endosome is acidic or it's lower. This results in a conformational change in the heme agglutinin or HA of the influenza virus. This will expose a fusion peptide, and once the fusion peptide is being exposed, as this one results in the fusion of viral envelope with uh, endosomal envelope, resulting in the release of capsid and viral nucleic acid into the host cell plasma membrane, host cell cytoplasm. So, through receptor mediated endocytosis, influenza virus gains entry into the host cell in an endosome. Endosome has acidic pH, results in a conformational change and exposure of a fusion peptide. This then there would be fusion of the viral envelope with the envelope of the endosome. The two membranes will fuse together. A pore is created through which viral capsid and nucleic acid will be released into the host cell cytoplasm. And this is basically slide number 22, a depiction of what we just discussed, the virus through fusion to the host cell plasma membrane gains entry into the endosome. Through an ATP dependent manner, hydrogen ions will be pumped into the endosome. This acidic pH results in the exposure of a fusion peptide. This fusion peptide, shown in red here, will create the kind of helps to uh, fuse viral envelope with the membrane or the envelope of the endosome, resulting in the release of viral nucleic acid as well as the capsid proteins into the host cell cytoplasm, and then viral nucleic acids through nuclear pores will gain entry into the host cell nucleus. Slide number 22 is a depiction of penetration of a toga virus via receptor mediated endocytosis. So, this is toga virus in figure part A. Binds to the receptors, invagination, gets into the endosomes. Slide number 25. After viral genome gets into the host cell, starts synthesizing early gene expression. As I mentioned, the initial step is the release of viral genome into either the host cell cytoplasm or host cell nucleus. Once the virus is inside of the host, it regulates, or my opinion, a better word is hijacks host cell metabolic machinery, and as a result of this hijacking, some viral genes are being expressed. These viral genes are expressed as a result of transcription of early messenger RNAs and early translation of those messenger RNAs. So, the result of this early translation is the production of viral regulatory proteins and enzymes that are required for the next phase, which is genome replication, as shown in Slide 26. Genome replication is the replication of viral nucleic acid. It uses either host cell or viral own polymerase. And once genome is being replicated, it sends signals that are required for additional viral synthetic effects and events. And these additional viral synthetic events are basically called late gene expression, which is further expression of viral genome resulting in late mRNA transcription and late mRNA translation. This results in the production of some regulatory proteins, but mainly structural proteins, those proteins that form the capsid and envelope. And these are proteins that are necessary for the assembly of progeny viruses. The next phase is assembly or maturation. This phase of viral replication, meaning assembly or maturation, is fundamentally different from microorganisms. Viruses are assembled from components or parts such as enzymes and capsid proteins, regulatory proteins, genome. And they are not assembled from division of a pre-existing virus. In other words, in viruses, unlike bacteria, we do not have an exponential growth kinetics, but we have bursts of new virions. And viruses that infect bacteria are called bacteriophages. So viral particles are being made, and when they want to assemble or mature, this assembly occurs from components or parts and not from pre-existing viral particles. Sometimes viruses undergo self-assembly. It's when the concentration of viral structural proteins and genome. These are the components that react with each other, so we call them reactants. When the concentration of these components are adequate, viruses undergo through a self-forming process when there is a recognition between various viral components, structural proteins, and genome. And this assembly follows basic laws of thermodynamics. For those of you who may have forgotten the laws of thermodynamics, there is a little bit of a description of it on slide 30 for just to refresh your memory. Slide number 30, virion assembly. Assembly of the viral particles requires protein and protein nucleic acid interactions. And these interactions are very, very specific. And the order that these interactions and assembly happens is via two ways. One is when the genome serves as a focus for assembly of the capsids surrounding it, and this results in the formation of helical viruses, or sometimes in as seen in icosahedral viruses, a hollow capsid is being formed and then it's filled with the genome. So let's see assembly of a helical virus such as TMV, tobacco mosaic virus. It results in the formation of a rigid helical virus, and which is made up of RNA plus identical capsimeres, or those components of the capsid that are arranged in a helix. TMV capsid proteins only recognize TMV RNA. So the interaction between the nucleic acid and the capsid proteins is very specific and unique. Slide number 33. We have discussed this before, assembly of TMV, the protein part of it. Initially, 34 identical capsid proteins assemble into a couple of discs. This is the formation. The outer portion interact to hold these two discs together, and the inner portion has a gap or lock washer, the type of structure that RNA will bind to. So the outer portion of these two initial capsids allows the capsids to be held together, and the inner portion of these capsids would be a region where there's a gap and RNA binds. And when the RNA enters into this structure, the gap is closed, and that will hold RNA in its place. So let's talk a little bit about the TMV assembly of its genome. RNA of the virus interacts with the discs at a region called PAC site, or packaging signal site. This PAC site is about 1,000 bases away from the 3' end of a genome. So about 1 kb downstream of the 3' end of a genome, there is a region in the viral RNA called PAC, or packaging signal, that interacts with the discs. And this PAC site itself is about half a kb, or about 500 base pairs, and it can form a series of hairpin loops. So for those individuals who may have forgotten what is a hairpin loop, I have included a definition of hairpin loop in slide number 35. A hairpin loop is an unpaired loop of mRNA that will be created when mRNA strand folds over itself and form base pairs with another section of itself. And the resulting structure looks like a U. So I would strongly encourage individuals who may have forgotten about hairpin loop to take a look at slide number 35 and try to refresh their memories. Okay, the TMV assembly, initially identical capsimeres associate with each other. See, these are individual capsimeres. They associate with each other to form a disc structure. And a couple of these structures will assemble, will sit on top of each other. The outside proteins will help to hold this two discs together, whereas the inside will basically forms a lock washer structure. Then the PAC site of the RNA will interact with this gap or lock washer structure, and this will result in RNA being hooked and Further discs are being assembled and will sit on top of each other. And at this time, the RNA will be threaded in through the central hole, and it forms a helical elongation. The composition of virus, RNA, and the capsid proteins form a complete virion. Let's talk about assembly of icosahedral viruses such as poliovirus. Icosahedral has 20 triangle surfaces. This is one surface. And each face of this triangle is composed of 3 subunits or multiples of 3, 6, 9, etc. These subunits can be identical or in some instances different. But usually, as we can see, they're all identical in this example. All are A proteins. So poliovirus assembly of the proteins and genome, initially protomer is being made, which is composed of VP0, VP1, and VP3. This will mature, and 5 of these protomers will come together to form a pentamer, or the end pentamer. 12 pentamers, 12 times 5 would be 60. 12 pentamers will combine to form an empty procaspid. This procaspid is when RNA will enter the procaspid. At the same time, there would be maturation cleavage of VP0 into VP2 and 4. So the complete virion contains VP2, VP4, VP1, VP3, and the RNA. So let's take a look at the assembly of the proteins and genomes. There are a bunch of proteins called coat proteins or capsid proteins. We have other types of proteins such as pilot proteins, portal proteins, scaffolding proteins, and at this stage, it's called an immature capsid or a procapsid. And once the genome will be inserted or added into the capsid through the portal protein, it was called mature virion or uh, a mature viral particle that is ready for being released from the host cell. Okay, so up to now we talked about how viruses gain entry into the host cell, uncoat, and then some uh, concepts about genome replication, early and late mRNA expression and translation, as well as virus assembly and release. So let's see what will happen when a virus actually infects a cell. Virus infects a cell and causes something called cytopathology. Cyto, cell, pathology, disease causing or damages. So virus lytic infections, those viruses that burst the cell and get out of the cell, cause very distinct changes within the infected cells. And these distinct changes are called cytopathic effects or CPE. And these changes include inclusion body, nuclear pycnosis or shrinking of the nucleus, formation of vacuoles, apoptosis or programmed cell death, and syncytia or the formation of multinucleated cells. Let's talk about some of these cytopathic effects. The first one, slide number 41, is inclusion or Negri body, as we can see in rabies infection. Slide number 2 is another cytopathic effect, which is uh, multinucleated cell formation, or syncytia, or giant cell formation. And syncytia or giant cell is one cell with many nucleuses, is seen during herpes virus infection. Upon maturation, virus will be released from the host cell. Let's talk about a process whereby host cell will be lyzed upon release of the virus. So cytopathic effects are usually a secondary result of changes in host cell metabolism that are dictated by viral replication. Viruses may completely stop or modify host cell DNA synthesis, its transcription, and translation or protein synthesis within the host cell. And all of these changes induced by the virus results in the disintegration of host cell and release of progeny viruses from the virally infected cell. So let's see another method of releasing of the virus from the host cell is a process called budding, when virus in the host cell endoplasmic reticulum, or Golgi, will synthesize a series of viral glycoproteins. These are proteins that undergo glycosylation in rough endoplasmic reticulum. Then virus will insert these viral glycoproteins in the host cell membrane. It can also insert them in the a host cell a nuclear membrane, or Golgi of the host, but it's usually, if it's getting out of the cell through budding, it is in host cell plasma membrane. At the same time, there would be the assembly of viral nucleocapsid, the nucleic acid of the virus that has been produced a lot through replication, and the capsid proteins that are a result of late gene expression. Viral nuclear capsid then recognizes that the region of the host cell plasma membrane that has been modified by the insertion of viral glycoprotein. So virus recognizes this region, says, oh, this glycoprotein is mine. It's not part of the host. So the capsid proteins, either directly or indirectly through a matrix protein, will interact with the virally modified plasma membrane, and then through a process called exocytosis, or budding, virus will exit the host cell, and the process of budding may or may not kill the host cell, whereas the process of cell lysis will definitely kill the host cell. Slide number 46 talks about polarized cell plasma membrane exit. Virus envelope proteins contain two types of signals: membrane transport signals. These are regions within these proteins that dictates them to exit either through apical portion of the host cell or through basolateral. So, viral envelope proteins have apical or basolateral plasma membrane transport signals. Those viruses that exit apically or through the outer surface, such as orthomyxoviridae and paramyxoviridae, usually cause localized infections. These viruses, orthomyxoviridae, paramyxoviridae, exit the host cell through its apical surface or outer surface. Whereas those viruses that bud out of the host cell through its basolateral uh, region tend to cause systemic infections. So apical exit, localized infection, basolateral exit, causes systemic infection. Viruses that exit basolaterally are called alpha virus, rhabdovirus, and HIV. These are just some examples of them. Slide number 47 is a definition of the basolateral cell membrane. As the name implies, basal and lateral is basically a fraction of the plasma membrane that is at the basolateral side of the cell, bottom and side. Basal, bottom, lateral side, which, as it says here, faces adjacent cells and the underlying connective tissue. So, neighboring cells and below the connective tissue, below the infected cell. So, the summary of the lecture: the events occurring during viral replication. Uh, they have been studied in detail in cell culture, and they are described as A, attachment and entry. Virus ligand recognizes specifically and binds to its host cell receptor. After this specific recognition and interaction between ligand and receptor, virus is taken into the cell where its genome is being released from viral capsid. The process is called uncoding. Then we have early gene expression where some viral genes are turned on and transcribed as a result of production of early messenger RNA. These early proteins are needed for regulation and viral nucleic acid synthesis. So these are regulatory proteins and enzymes. Then we have genome replication, where is the synthesis of virus, nucleic acid, and the production of many, many copies of viral genome. After which we have late gene expression, where the remaining virus genes are turned on, transcribed into late messenger RNA, and late proteins are being produced, and these are the proteins are being produced by the translation of late messenger RNA are usually structural proteins, those proteins that participate in the formation of viral capsid. Lastly, we have assembly and release, which is the recognition of virus nucleic acid and proteins. When nucleic acid and proteins, in a very, very specific manner, following the laws of thermodynamics, recognize each other, this and if their concentration is adequate, results in self assembly of virions. So we have viral proteins being made, viral nucleic acid is being made. We have many copies of viral nucleic acid, lots of capsid proteins. They assemble through specific recognition of virus nucleic acid and its proteins, self-assembly and the formation of viral particles or virions. These are new viruses or progeny viruses that they will be released from the host cell either through cell lysis, which results in the death of the virally infected cell or the host cell, or through budding. Budding may or may not kill the host cell, but results in a piece of host cell plasma membrane being wrapped around the new viral progeny as it exits the host cell, and this virus becomes enveloped virus. The reading material for this set of lecture slides comes from chapter 6 of the textbook. Thank you very much for your attention. This is the end of lecture on viral replication. Thanks.